All right, guys, I am going to be going over the ear today, the histology of it, which I'm going to be straight up with you. This is a lot more like anatomy, but, I don't care. We need to get good grades on these quizzes, so it doesn't really matter to me. So, before I even go into any of the structures of the ear, I'm going to start off actually by highlighting these two specific terms that we are going to be using an annoying amount during this video. So these two terms are going to be endolymph and perilymph. Now, what's significant about these? So, these are types of fluid that are kept in the different parts of the ear. And if you don't know, now you're going to figure it out. A lot of your balance and equilibrium is actually determined by the fluids in your ear and the way they interact with the ear anatomy. And the stereocilia, the little hairs that line your ear, whatever they come into contact with, whatever direction, it all is based obviously on, the way you're facing, whether you're shaking your head around or going up and down. The fluid is going to pick up on that movement and it's going to act accordingly. So going into that, the endolymph is for fluid for the membranous labyrinth. I always misspell that. And the perilymph is for the fluid for the bony labyrinth. Now, something to note that's a little significant about the perilymph that gets mentioned enough to where it stresses me out that it might be on the quiz, but better safe than sorry. The perilymph fluid is similar in concentration to cerebrospinal fluid. And it's high in sodium ions. Don't ask me why. I don't really give a shit why that is the way it is. I just know that we might need to know it. And then for the endolymph, that's a really shitty arrow. Sorry, I'm OCD. The endolymph fluid is consequently high in potassium ions. If you think back to our sodium-potassium pumps and how they work in conjunction with each other when it comes to polarization and repolarization and depolarization events, you can see that these fluids working, respectively to one another, it kind of ties into that action potential theory. And now that we have these mentioned, we are going to talk about the different parts of the ear. So you have got three main parts. They're very, very creatively described. So the first creative description, the outside, is going to be the external part of the ear. And then the middle is going to be, I'll let you guess for a second. It's going to be the middle. Yep, I didn't know if you were going to get that one. The middle part of the ear. And the final innermost part of the ear, wait for it. This is the most creative one, actually. It's going to be the internal part of the ear. Now, going in order from external to internal, we're going to start off with the external portion. Now, the external portion starts off with the auricle. You remember that term that we learned in anatomy with the part of the heart that is called the auricle, and it looks like an ear? Well, this is the auricle of the ear. So, some next level shit. And so the auricle is cartilaginous. If you need me to explain what that means, you should drop out. And it directs sound vibrations, which we're going to abbreviate from now on as SV, because I'm lazy, to the external acoustic meatus. Now that's the first part of the auricle. The external part of the ear. After the auricle comes the external acoustic meatus. And this part, it's a lot like the auricle, except it also has a secondary function. It again directs the sound vibrations to the tympanic membrane. Tympanic. I misspelled that. Whoops. Tympanic membrane, which is one of the things that the professor told us to specifically focus on. Wink, wink. And the secondary function of this is it actually contains ceraminous and sebaceous glands, which clean your teeth out. Now, finally, for the external ear, we're going to have the tympanic membrane. Man, I'm not having any luck with these arrows today. So this is basically your eardrum. The tympanic membrane is the scientific mumbo-jumbo jargon for the eardrum. It's your eardrum. So whenever you hear tympanic membrane, like a timpani, like the instrument, just think you're slamming the timpani. So you're slamming an eardrum. It's an eardrum. It's an instrument. So it's kind of like a thin cone-like. It's like a thin cone-shaped membrane. If you know acoustic physics, cones can propagate sound better than most shapes. So it does have a little bit of biophysics in that as well. Luckily, we're not taking that. And it receives sound vibrations from the external acoustic meatus, and it transmits them to the ossicles of the middle ear. Now, a little Latin lesson for you. Os, whenever you see that, 9.9 times out of 10, it's going to be referring to bone, because that is the Latin prefix for bone-type material. So in that case, take a guess as to what the ossicles of the middle ear are. So the ossicles are the 3 tiny bones inside your ear, And for some reason, they decided to equate these bones to blacksmithing terms for horse equipment. I don't know why, but again, and I can't stress this enough, I could not give less of a shit. So onwards we go. 3 ossicle bones make up the middle portion of the ear, and the first one is the malleus. Now, this is also referred to as the hammer. And it's actually connected to the tympanic membrane. I'm going to abbreviate that as TM because I'm lazy. And it transmits sound vibrations from the TM, the tympanic membrane, to other ossicles. Now we've got the incus, not the Incas. That is a Native American tribe. I don't know how much y'all know about that. It's a very American thing. It's basically the kind of brown but not quite people who chuck spears. Those are Native Americans. Those are Incans. But we're talking about the Incas, the anvil ossicle bone, not the tribe. And they receive sound vibrations from the malleus and they transmit the SVs, the sound vibrations, to the stapes. Now the stapes are called the stirrup. And they transmit the SV, the sound vibration that has come all this way. The previous step being the incus, to the oval window, which is part of the inner ear. Now, the internal ear, it can't just be listed just like that. It's a little bit more in depth, but the main parts of it are going to be the cochlea and the vestibular system. Oh, and really quickly, all these 3 ossicle bones, they're housed in the tympanic cavity, your ear canal, basically. Now, the internal is how it's comprised of the cochlea and the vestibular system. Now, if we were to go to a diagram of such, as you can see, it looks nothing like a hammer, anvil, or stirrup. But, again, we don't get paid enough to care about that, so we're just going to roll with it. So here, this portion, these 3 things that it's pointing to, these are the semicircular ducts. And these semicircular ducts are actually split up into 3 different portions based on their anatomical position. So, it does that. Topmost being the anterior portion, the anterior semicircular duct. Middle one being the lateral. and then the bottom one being the posterior. And then on the other side, and this specific one that I'm about to write down is going to come into significance a lot more later, so stay tuned for that. This is going to be the cristae, and every single semicircular duct has one of these. It has a cristae that's within ampullae. So these are the ampullae, and the cristae is inside them. So it's housed within the ampullae. Now, this is going to be the utricle of the ear, also going to come into significance later, and the saccule of the ear, also going to come into significance later. And how, like all these impulses, all this information, like it's, these are sensory stimuli. Like they got to go somewhere, right? There's got to be something that catches them all up. So this bad baby, the yellow might have given it off, but this is going to be the vestibulo-cochlear nerve. And this seashell, Gary's shell looking thing, this entire thing is actually going to be the cochlea. And the bottommost portion of it is going to be the tympanic duct. The middle portion of it, where the blue is, that's going to be the cochlear duct. And this ribbed portion is going to be the vestibular duct. Now, remember the terms that I mentioned in the beginning? The ones that I said were going to come up a lot later on? Now, they're about to. Before we even start, just so you can differentiate them, all the purple that you see in here, that is going to be fluid filled based off of the bony labyrinth. And the bluer portions, that is going to be fluid based off of the membranous. Bro, I'm going to be really upset. Membranous labyrinth. Cool. So now that we have the anatomy set, I'm actually going to let you look at it for 5 more seconds. We can start off with the cochlea and start explaining that, because it does get a little in-depth. The cochlea. Gotta love the names they come up with. Don't know why they named that. That's beyond me. The cochlea itself actually gets segmented into 3 separate layers. I'm going to do that one more time, because OCD. 3 separate layers. Now, you've got the lowermost layer being the scala tympani. I'm just going to label that real quick just for y'all's reference. And then you've got the middle portion, which is labeled rightly so, the scala media. And the uppermost portion, someone's about to get arrested. Don't mind that outside. Scala vestibuli is going to be the uppermost portion of the cochlea. Now, these layers actually, both the layers, besides the media, the tympani and the vestibuli, they intersect with the media and have certain membranous attachments that connect them between one another. So the one that connects the scala tympani and the scala media is going to be the basilar lamina. And the one that connects the scala media to the scala vestibuli is going to be the reticular lamina. And these bad boys are joined all together onto the scala media by our friends, the rods of Corti. Now, the cochlea, with its layers, we're going to go into the layers in depth a little bit. The scala, let's bring that a bit down. The scala tympani, so this is filled with perilymph. A quick gather back to what we said. Perilymph, it's for the bony labyrinth, remember? So this is going to be filled with perilymph, which is for the bony labyrinth, and it's the one that is very similar in concentration to cerebrospinal fluid. And it connects to the oval window. Now, the scala media is a little high maintenance and thinks it's really important, so it's got a little bit more going on than the other ones. So this one is filled with endolymph, which is the membranous labyrinth fluids. Again, all of these, like the ear is based in its equilibrium standpoints off of fluid. You got to remember that. That's pretty significant. And the scala media houses the organ. I'm actually going to write this in red because the organ of Corti is actually pretty significant. The organ of Corti is the main structure for... I'm actually going to write a little bit of a dash to that just so you know it's specifically talking about the organ of Corti. It's the main structure for the sense of hearing. Now we're going to go over to the organ of Corti diagram here, and we're going to fill this out. So you've got the tectorial membrane at the top of the organ of Corti, and this extension right here is going to be your inner hair cell. As you can see, it's contained within the matrix. And then these little bundles right here, they're going to be hair bundles. And the reticular lamina, just like you saw right here, reticular lamina is the one that separates the scala media from the scala vestibuli, and that's kind of what is happening right here. This membrane, it's separating the separate layers of the cochlea. So this is going to be the reticular lamina. And just like you saw this was a cell. It's got this little nucleus right here. These are also cells with a nucleus. So if this one's the inner hair cell, this one would, by the associative property, be the outer hair cell. And this purple mess right here, that is going to be the basilar membrane. Okay, and that's the, I'm actually going to go back one more time and let y'all look at it for a little bit. That is the organ of Corti. Now, humans, we have a specific range of hertz that we can listen to or, conceive sound from. Hertz is the unit of measurement that is used for wavelengths of sound and the way they propagate in conjunction with our cochlear anatomy. So 20 hertz is the lowest frequency noise that we can hear. And 20,000 hertz is the highest frequency that we can hear. And of course, as you saw in the diagram, the organ of Corti comprised of basilar and reticular lamina. So specifically for the scala media, it's kind of tying into the the frequencies that your ear can pick up. Hair that is specifically, the hair cells that are specifically close to the oval window, they tend to be stiffer and shorter in length. And this results in them being able to pick up high frequency sounds. Whereas hair cells that are close to the end of the cochlea they're looser and longer, resulting in them being able to pick up the opposite, low frequency sounds. And now moving on to the scala vestibuli, the uppermost layer of the cochlea. It's pretty simple. It's very similar to the scala tympani. These kind of both house the scala media. It's kind of like their outer borders. The scala media is the most important part. These, they help with sound transmission and propagation of sound volume waves, the sound vibration waves. But at the end of the day, out of the scala tympani, scala media, and scala vestibuli, the scala media is going to be the most significant one. So the scala vestibuli, just like the scala tympani, is going to be filled with perilymph, which is the fluid based off of the bony labyrinth, as we talked about earlier, and it also actually connects to the oval window. Oh, really quickly, the scalum media is also sometimes referred to as the cochlear duct. So if you see cochlear duct, it's just the scalum media. Now, there are two parts of the ear for the internal ear. There's the cochlea and the vestibular system. We just covered the cochlea. Now we're going to go to the vestibular system or the vestibule. Either works. I just hope the professors think the same way. I can't really tell. But it could be the vestibular system or the vestibule. Now, the vestibule itself, from a macro standpoint, is going to be filled with perilymph, the fluid based off of the bony labyrinth. I'm gonna keep repeating that, even though it's redundant, so it sticks in your head and my head too. Because I remember I said this is for my studying as well as it is for yours. And as I mentioned earlier, I was going to say that the utricle and the saccule are going to be important. The vestibule is filled with perilymph, and it's composed of, what do you know? The saccule and the utricle. Now, these both actually have a pretty significant function respective to each other. They do very similar things, but it's just the directions of what they do that's significant. So when we're talking about the saccule and the utricle, anatomically, the saccule is actually going to be inferior to the utricle. The utricle is going to be a superior anatomical structure to the saccule. And what these two things do is that they support static equilibrium. So that's a lot of words and letters that basically just mean that the saccule and utricle, they basically monitor whether or not you are going forward or backwards, like up and down, horizontal versus, lateral, or horizontal, lateral versus vertical movement. So they both actually respectively do those things. So if it's horizontal movement, kind of like a car going side to side, that is going to be focused on by the utricle. And the vertical movement, which is kind of like if you're going down a roller coaster and it goes insane and you get that feeling where you're like, oh my God, I'm going down super, super fast. That's going to be basically your saccule going into overload mode, because that's what it tracks. And so that brings us down to another part of the vestibule, as you saw, the semicircular ducts and the canals. So the canals are actually what houses the ducts. So if we were to start off from the most superior structure, we're going to start off with the canal. Semicircular canal. So the semicircular canal is going to be filled with perilymph. which is the bony labyrinth-based fluid. And there are 3 portions, as you saw in the anatomical depiction of the internal ear. There's 3 portions: anterior, lateral, and posterior. So we've got the anterior, lateral, and posterior portions of the semicircular canal. And now, each semicircular canal, SCC, it contains bulbs called ampullae. I told you that was going to be more important. I told you it was going to be important, the ampullae. Now, these ampullae are the sensory part of the canals. And these ampullae, as the diagram showed, they contain cristae ampullaris, which is the full name. And basically what these house are the hair cells. It all boils down to that simple thing. It's like the binary code of biology. And like I said, the semicircular canal houses the semicircular ducts. And these are filled with endolymph, which is the membranous labyrinth-based fluid. And that's pretty much the ear histology, but she did mention that there was something that she wanted us to know for the quiz, which was otitis media. Now, this isn't anatomy. I just want to make that very clear. This is actually an infection. You want to always remember the suffix itis. Whenever you see itis, it's almost always going to refer to inflammation. So, otitis media, the media part, it's an ear infection. What part? It's a middle. Oh, that was weird. It's a middle ear infection. It's inflammatory. And severe cases of it require surgical insertion of a, I'm going to use a smaller pen for this because it's a long word, require surgical insertion of a tympanostomy tube. So basically, otitis media is a middle ear infection. It's inflammatory, and if your case gets severe enough, it's going to require the surgical insertion of a tympanostomy tube. And that, my friends, I'm going to go back to the anatomy of the internal ear and the organ of Corti, just so you guys can get a picture of it. But that is pretty much the histology of the ear. I just went over the surface level parts that we've seen her take the quiz on, because I guess the deep stuff is for the final. But there you go. Let's get some 10s tomorrow.